imaging cytometry, single cell RNA fish, MERFISH, multiplex barcoding technologies that allow us to assay RNA variation in hundreds of markers. And these technologies essentially have in common that we're now able to measure expression variation in a 2D coordinate system, that we don't measure cells that are dissociated, disordered, where we don't know which cell is where, but essentially retain the dependencies between them. And there's a number of interesting differences one could mention about these technologies, which I'm not going to do massively into detail here, but I just want to highlight there are trade-offs everywhere, and it's still, I think, an exciting domain to understand which technology is best for which question. The two axes I'm showing here is the number of reported genes. So is this a genome-wide method? Spatial transcriptomics, for example, the latest Visium technology from 10x, Fawson's technology is genome-wide, transcriptome-wide, 10,000 genes that we can assay. Other technologies like MERFISH are not transcriptome-wide, hundreds of genes, but maybe have more throughput, larger numbers of cells. And there's a third dimension which I haven't shown, at least one other third, which is sensitivity. The more markers you measure at the same time, the less sensitive typically these assays are. That's a common rule of thumb. And I should also mention that some of these methods I'm highlighting here, actually, spatial transcriptomics from Visium, for example, is actually not really truly single cell. It's 50 cells. Maybe we can push this down to 5 cells, but it's a voxelized method. So readout is a group of cells that are close in space, but it's not at this point in time a true single cell. And I think it gives you a sense of that there are differences in technologies and others. So a few years ago, we thought about, what are the intrinsic problems though, however, and I think this is quite exciting to think about because we essentially solving a different modeling question than previously. We're measuring now the abundance of a gene, say, let's say this is one gene, and every one of the little towers here is representing one cell or one voxel, and we're measuring the abundance of that given gene at these different locations, and some are high and some are low. But it becomes very intrinsically important to interpret this variation in the context of this coordinate system and ask one very simple question, namely, does space dictate gene expression variation? And that sounds a bit trivial, almost like a boring question, because you would mention, well, space dictates all expression variation, but it turns out it actually doesn't. And it's interesting to see which genes vary in the spatial way. Those which do are mostly also tagging tissue histology and biological processes of interest. So understanding which genes look like this versus other contexts of variation where spatial variation is independent a priori of expression is something interesting. So the approach we took for that is essentially modeling these gene expression data using ideas from geostatistics. That's at least one of the many communities that has engaged in these models. Others, they may be known as random effect models or Gaussian processes. So the key idea is to essentially model the covariance of a single gene. So Y here is a vector of one gene using a random effect model, one covariance matrix. And this covariance matrix here measures how distant are, well, actually how similar are the locations of two cells from the same gene. So basically, this sort of diagonal stripe that you see here in this matrix here, these are all voxels that are nearby. And essentially, you're asking whether the spatial covariance, the similarity in space that is represented or captured by this choice of covariance or kernel, is predictive of gene expression variation. Or put it otherwise, are voxels that are nearby similar in expression space or not? And obviously, you can think about distances. There are a number of distances, a simple distance, periodic distances, or something we've been playing with, or linear trends. But this is the basic framework, basic setting. It's a very simple and straightforward idea. I just want to show you very briefly two results. So we applied this to a number of data sets. Here is a mouse olfactory bulb. It's sort of a nice histology data set with two bulbs, and you see this is the histology image. So this is 1D. Basically, here you see marked cells and locations, DNA content. And then we applied this approach and basically ask, which genes are spatially variable. This is the output on the x-axis, the fraction of variance explainable by space. So things on the right are more driven space. On the left, spatial position is less predictive of gene expression. And you can also make a statistical test and ask which of these variations are significant. And there's a number of them. It's 70 of them. And I think actually 70 was quite a low number. Given the structure of this tissue. And that's something we see actually in a number of data sets. So filtering by space, by spatial variation, is a very interesting, or at least a useful first-line analysis approach for understanding which genes are governed by such spatial differences. This is actually the spatial variable genes that you identify. Here you see the actual resolution of the assay. So every dot here is a voxel. That's what you're measuring. And you can see some of those genes with nice spatial patterns. I show you 6 of those. We could obviously just look at all 68. It's somewhat unsatisfying, though, to do that, because you see clearly similarity. For example, this gene PN key is very similar to NRGN in spatial patterns. So the next question was, can we bring those together? And I just want to be brief here. Yes, you can obviously do this. You can use these ideas of having a smooth spatial prior on expression levels and also generalized clustering. So here, the same idea. We have a covariance model, GP model, that encodes the assumption that loci or voxels that are nearby are characterized by similar expression profile. That's a prior. And what you then essentially do is with the k-means clustering, you're assigning genes to these reference profiles, update these profiles, and reassign those genes, and but accounting for the spatial prior, and thereby given essentially spatial prototypes, of different expression programs that you encounter in the tissue, and you can look at, and and this is sort of how this looked like. It's a quite straightforward idea. You essentially decomposing the 68 genes into a number of major groups. In this case, we see 5 groups, and there is a group where genes are expressed in the inside, and there's a group where genes are expressed in the outside, and other elements. So I think it's just sort of giving you one new dimension. Yes, there was a question
in the chat. So Will Noble asked, for the mouse olfactory bulb analysis, do you have a sense for how good your power is? Because 68 is a small number because there are few such genes.
Yeah, it's a good question.
Test as low power.
Yeah, that's a good question. I mean, there've been some extensions that actually very similar to spatially. We also have a different model power. I mean, the 2 elements. One is the test itself, and there are some tweaks you can do that increase power. I mean, I think increases of 30, 40, 50% are certainly viable. It's still a small number in relative to the whole transcriptome. I think that's biologically true. The other elements, the assays become more sensitive. I mean, with later assays, there are clearly power increases where you do this. But it's a good point, that you have to sort of take with this grain of salt that these assays remain also limited by sensitivity, and that is certainly one element. But the substantial variation that's just not driven by space. I mean, many housekeeping genes just do not vary in a spatially coherent way. And that's also something that will remain, is a generic observation in these types of data. Yeah. Okay. Then I would proceed. I mean, the other dimension. I mean, this is just a bit of walkthrough of sort of different dimensions of single cell variation, where I'm trying to sort of align these more broadly themes. The other, I think, very exciting trend is that we are now increasingly able to measure variation in cells at the multi-omics level. And that's sort of complementary. So the previous chapter was measuring RNA or protein abundance with single omics in space. The other dimension is looking at multiple omics layers in parallel, but from conventional disassociated cells. And this is a quite active field of still development. At the moment, these assays are quite bespoke, by and large. So we worked for a long time with Wolf Reichslab and had a series of protocol advances of making that happen. I'm just showing here the picture of SC-NMT-seq, which provides nucleotome methylation and transcriptome sequencing from the same cells. The protocol that physically separates the RNA from the DNA and is enzymatic by SOFA treatment based for assessing both methylation, which is CpG methylation, but also nucleon positioning via GPC methylation. And but it's just one representative of many similar assays. What's a nice advantage about this assay is because it's by sulfide based, you know what you don't know and you don't know. So missing values, in the sense, at least in the epigenome side, are well identified, because the information sits in the conversion, the bisulfide conversion, and not the observation of reads. But now there are also other methods, I think, that using tax-based sequencing methods that combine this with the multi-omics dimension, and and go forward. So I mentioned before that matrix decomposition is sort of a generic, I think, toolbox that remains very applicable to single cell assays. One approach we've been, I think, also been pleasantly surprised by how far you can push this is a group factor analysis model. So essentially, instead of decomposing a single matrix, as illustrated earlier, you should just do this at the same time with multiple of these omics layers. You know, let's here metallone, genome, proteome. It doesn't really matter what they are. You're making here the assumptions, however, and that's an important one, that you observe these different omics layers for the same samples. Yeah? So you really have a true multi-omics observation where different assays are measured in parallel from the same physical cells, which is obviously only possible using such bespoke assays that generate a genuine and multi-omics data. What these sort of group factor analysis type methods allow you to do, and that has been a very, I think, fruitful endeavor, is to basically generalize these variance plots. You know, you're asking which factor explains how much variance, but now you can ask which factors explains how much variance in which assay, and in which assay at the same time versus, you know, as being specific to some assay. And I'll show you later on application of that. And I think that's a quite nice way because we can also start making connections. We can, you know, annotate that specific sources of variation are coupling two assays and potentially link them to a particular biological process. And I think this is an interesting way to draw connections or discover variation in these types of frameworks. So for those already, you know, been playing with MOFA and looked at it, there's just a one slide plug. There's also a second version of MOFA where we generalize this now also not in views, but also groups. So we have different groups of cells and different modalities. So you can also account for the fact of different cell types and cell states. And I think it's now quite a generic framework that's very fast and flexible for dealing with a number of sort of generic single cell assays and questions. And we can use those here. Here today, in the interest of time, I want to just you know, show you one use case, and I think it's just speaks to the power of these multi-omics methods almost more than the specific analysis. So here with Wolf's lab, we looked at mouse gastrulation, the early stages of life from E4.0 to E7.5, so very early stages of gastrulation, and performed and characterized these transitions using single cell nucleotide methylation transcriptome sequencing, where we basically obtain coupled RNA methylation and DNA accessibility profiles from these cell samples, roughly 1,000 cells or so. So a small data set, and what you can already see is that the transcriptome is extremely predictive of these different stages, massive changes. The signatures and methylation accessibility are in general much less obvious and much more subtle, and require a bit more thought how to extract those. That's maybe one of the sort of messages that, or lessons we learned from doing these types of analysis. So what we're doing here is now apply this MOFA model, and essentially what the rows are different factors. So these are basically 6 major components. I'm only showing 6, and there are in total 20 or so. They've also names, and these names actually come from the RNA view. So from the RNA, we can look at what are the genes that are mostly associated with one factor, that's essentially genes of enrichment. We can identify one factor is linked to mesodermal lineaging, another one to endodermal lineaging, axal patterning, you know, but also sort of intrinsic processes like the cell cycle are extremely present and prevalent and have always been very prevalent in dividing cell populations. Now, what the columns show are the different omics layers, chromatin accessibility, methylation, RNA, but we then also subdivide the annotation, the assays by further annotation. So what we actually use here is we use annotations to derive lineage-specific enhancer marks, for example, distal H3K27 acetylation events, or promoter marks, that are by carrying out ChIP-seq in these specific lineages. And then essentially what you see here is sort of dividing the genomic space of CpG or GPC methylation into these categories, yeah, and assessing essentially how much variation does this one factor, this joint factor, explain in CpG or GPC methylation in these specific areas. And what this revealed is was quite striking to us is a number of, I mean, some are expected patterns. One of them is that in early development, promoter methylation explains almost no variation that's specific to development, very well known, and we observed it also very strikingly, but the actions, it's actually distal enhancer elements, and they're lineage specific, which is also, I think, quite nice to see that endodermal enhancer variation are linked to endodermal enhancers. We could also see that there's a default commitment layer linked to ectoderm. And then you can obviously also look at what do these factors look like, and how do they start separating these lineages. Actually, this sort of simple view was the major hypothesis generator for the biological follow-up and in-depth analysis, which I'm not presenting here today, but I think it just shows to the sort of power of explorative data analysis and simple good old fact analysis. I think is still a very, very useful tool for us. So I've tried to sort of just present some of the recent areas we've been encountering in new omics assays. But now I want to spend the second half of this talk to really go back to my slide 1 and show you some efforts, primarily methods that have been developing for scaling single cell variation, to population scale experiments. I mentioned that often we don't have the labels. Obviously, in the population scale experiment, you do have the labels. You can ask, what is different in single cell states between people, and that's now becoming feasible as we scale single cell genomics to large cohorts and large data sets, and that required some thinking about how to do this experimentally, but also how to set up the analysis for such work. So we've been doing this in the context of human iPS cells, and I just want to briefly bring some background, because most of this work is not single cell, it's in bulk. And I at least need to recap the conventional workflow of doing genetics analysis in bulk assays before I can go into single cell variation. It's a fantastic consortium based in the UK that has generated a huge biobank. It's actually an open biobank, and you can access all the lines and the material online, actually on HIPSCI.org. Also, most of the data which I'm presenting now is accessible. So for anyone excited about this, most of it's even open access, so no dbGaP application. You can just go there and download these data, which I think is very exciting and liberating in many ways. And we've now generated more than 800 iPS cell lines from more than 500 individuals. And we've characterized initially these iPS cells themselves before then also doing differentiation experiments, which is where I'm going to talk about the iPS, the single cell dimension. I just want to quickly mention how these conventional genetic analysis are conducted. I think for many of you that's no big news. Essentially, the number one approach for detecting genotype phenotype associations is to look locally in the vicinity of genes for regulatory variants. For example, here you see this variant, which is, I'll show you later where that variant is actually in intron of a gene. And those individuals carrying the DT allele have low expression, while those individuals carrying a CC allele have high expression. And you also see the heterozygous intermediates. And that's sort of the cartoon. This variant might, for example, act by affecting binding or other means of transcription factors. And that's sort of real data. So it is stratifying, and that's an important concept to take forward from here, to stratify the population by genetic state at one position in the genome, and then ask, do we find systematic differences in gene expression levels across people? And I mentioned, this is sort of one variant that was of interest. Here's actually the real eQTL. This is a gene TERT, is TERT reverse transcriptase. This gray bar here is the coding region of the gene. And you see here in intron 4, there's this one gene, this one variant, which is strongly associated. The y-axis shows you the minus log 10 p-value for association with expression changes of TERT. So here, these are variants where the eQTL have shown you before. And it's not just any random set of variants, it's actually variants that are also risk factors for prostate cancer and breast cancer. So, you could even argue that we've identified potentially a molecular mechanism of these cells. And this is in human iPSCs, which is perhaps surprising, perhaps not. It's essentially a system for pluripotent cells in human, and that this is maybe suitable for modeling risk factors in cancer is debatable, but I think it was a very exciting dimension because, for example, you don't find this in GTEx and anywhere else with a specific association. But this is a generic approach, and we've taken a number of different areas with this cohort and iPS cells. I just want to mention here one element, which is just scaling up. Yeah? We've meanwhile now not only like 500 iPS cell lines, but we have iPS cells collected from 1,000 people. This is work with Stephen Montgomery's lab and Kelly Fraser, and led by Mark-Jan Bonder and Craig Small, and there we have now more than 1,000 iPS cells, and you can see here where these iPSCs sits compared to GTEx, extremely homogeneous. And I highlight this and emphasize this also compared to later where we start differentiating those. And this is, you know, combining data from 4 studies. I think it shows you how actually well controlled the generation of iPS biology meanwhile works, that we can now establish. This is just, you know, how many genes do we find? We find meanwhile eQTL, so regulatory variants. This is for the individual cohorts. This is a mega analysis for almost the whole transcriptome, and this is actually quite a number compared to, again, GTEx tissues. It I think really speaks to the power of doing genetics in iPS cells. It's quite a, I think, surprising result how easy it is in a way to map genetic effects in iPSCs, because I think they are so homogeneous and well controlled. This is, by the way, a large blood eQTL mega analysis with a much bigger sample size. But we so basically know, you know, the regulatory landscape for pretty much every single gene in human in iPSCs. So I think we're really done with iPS cells. And now what we've been excited about is the opportunity, obviously, take these as a model and differentiate iPSCs into somatic cell types. And the advantages are clear. We have a renewable system. We can do the experiments over and over and over again. And we can also study, in a way, how human pluripotent cells exit from pluripotency, how they start to differentiate into other lineages, and what's the dynamics of this process, something you'll see more about later on. That's where the Ludovic Vallier's lab in the UK and John Marioni's lab, and it's really led by Maria Chatawala, lab manager of Ludovic, and students and postdocs in my group have been driving the analysis. And before going there, I just want to highlight, I think, an experimental design choice that is something that we are very excited about. I mean, we don't look at iPS cells one by one, but we look at them in pooled aggregate. So here you see 4 iPS cell lines. We disassociate them, and then we plant synthetic mosaics, so little colonies that now consist of cells from all 4 people. Now, you know, here it's 4. Actually, you can do much larger batches of 20 lines is even doable. And then we start differentiating these pools towards, in this case, endodermal lineages. So this is a model system we chose because endoderm is very well controlled. There's 72 hours of differentiation, so it's quite tractable. And you can use this and then do the single cell sequencing. And, you know, on the one hand, this sounds great because we obviously mitigate batch effect. You reduce also the experimental effort. But, you know, you have to also deconvolve these mixtures and understand, you know, which cell now came from which donor. Otherwise, obviously, the ability to do genetics is limited. You need to be able to reconstruct the labels, in this case, the genetic labels. And I would really sort of revisit this theme 1 of this talk. We need to figure out that label, that genetic labels from these cells. And this is actually now a whole industry. There's a whole number of methods available. I'm just showing you the approach that we developed. Essentially, what we're using is we use genetic variants to segregate between these cells. So you can, from the single cell RNA-seq, discover mutations. This is actually real data, SmartSeq2 data. You know, you have some power of detecting these changes. It's not perfect, but it's sufficiently good, clearly sufficient to de-identify individuals. You can even use this information to map cells onto somatic clones in aging tissue. So you're able to really reconstruct the cell of origin state from the single cell data quite well. And I just want to show you here basically what has been the key innovations of our approach. Essentially what we do is some sort of clustering of variants. So we essentially cluster cells not by their transcriptome state, but their allele specific expression state. So we ask, do we see evidence that at least one read maps onto the mutant variant, on the heterozygous variant that supports the presence of a specific mutation? Using a binomial overdispersed binomial noise model, but we also actually account for allelic imbalance we expect in genes. Not all genes express both alleles at the same rate, so you have to sort of account for this background model changes. And if you do so well enough, you know, you can start deconvolving quite complex mixtures. I just want to show you here one benchmark plot. You know, if you think about how many variants do you need that segregate between 2 variant branches, roughly 10 variants is what it takes to do these inferences. It's quite surprising how well this works actually in practice. Now, I want to go back to this picture and just convince you, I hope I convinced you, that taking these mixtures of cells from different people and assigning the label of cell of origin is an easy task. Obviously, between any two individuals, you have tens of thousands, 10,000 roughly variant segregating. So there's sufficient information in the coding transcriptome to do this with high accuracy. And now we can start sort of looking at the data. So this is sort of what we got. This is this blue population here. These are iPS cells. So these are iPSCs. Here I'm ignoring the label information. So this is from 127 donors, all pooled together. This light blue population, these are iPS cells on day one. So they basically accept pluripotency and are on the verge of differentiating towards endoderm. This is 2 days after differentiation. There's orange population, and in red, 3 days. And you can already see that after 2 days, some cells look like these red guys already. There's, this clear notion of differences in timing, that cells propagate down these processes with different speeds, that we can now reconstruct from the transcriptome. That's actually what we do. We use a very, very, very simple approach. We use the first principal component to reconstruct a pseudotemporal ordering and assign these guys here on the left, as early stages, and he's on the right as late stages, and essentially use this linear trajectory. And this is an easy problem because there's no branching. There's no real major decision points. It's pretty linear, except for these noisy cells in the middle, which are which we actually ignore in the analysis. They're mostly low quality. And now what you can do with this is you can essentially gate your cells in a data-driven way. I can define that all the cells up to this point here, call these iPS cells. These cells in the middle stages from here to here, we can call mesendoderm, and then here at the end stages, we can call these cells here definitive endoderm. And essentially what this allows you to do now is to summarize and pool the cells from these 2 or 3 different stages, and then go back to normal QTL mapping. Here's an eQTL. It's a variant in a gene promoter. The name of the gene, unfortunately, is missing. Apologies. This is my rushed copy and paste activity earlier today. But what you can see here is that in the iPS cells, if you take all these cells that are labeled iPS, those guys on the left, I don't see changes in expression of this gene, CNTRPA2. So it doesn't matter. Carriers of here, every dot here is not a cell, it's a person. So we're looking at the average gene expression of this gene by pooling across all the cells within a person that are gated as iPS. And now we look at differences between individuals as a function of their allelic state, and there's no effect neither in the intermediate stages, but we do see a strong effect in differentiated stages. And so we can basically, choose ourselves where to do this analysis. And, we can do this obviously for one gene, we can do it for many genes. There's a number of genes we find. We have a decent power for mapping QTLs, not the whole transcriptome, as I've shown you previously in this bulk data, but for 127 people, it's pretty reasonable. And what's also interesting to see, if you gate cells just by day of collection. So this blue bar here is just saying, let's take all the cells 1 day of collection, not looking in a data-driven way. I'm just quickly jumping back for this developmental stage defined by the pseudo time, but just looking at the time of collection, we have actually a lot less power for finding eQTLs. And that's also quite plausible because we don't gate for a homogeneous transcriptome state, but averaging over lots of things in the same bucket. Now, this is another gene, ZPZ, where here's again the coding region. We see here different variants. And what you see here is that this gene has 2 different eQTLs depending on in which stage you're assessing regulatory variants. One in the coding region in iPS cells, and in differentiation, these things change. In this case, we actually can link this to a change of enhancers. So it's quite a nice example where we can find a defendoderm specific enhancer that's only becoming active in the differentiated state, and that explains the change in regulation. Now, the exciting, I think, really area of this opportunity of these types of data is that we can map now dynamic effects. However, so we can, I mentioned before, we bucket cells in the different regimes, those early, middle, late, but it's very discrete. It feels ad hoc, and any such decision of gating is ad hoc. It is as ad hoc as a facts-based gating. You have to choose a marker, you have to make decisions. And we're obviously interested in, can we do this more dynamically? And what we do here is essentially, is ordering cells along the pseudotemporal ordering as a time component, and then ask, how does expression variation change along this component? So that's straightforward. But we can do the same ideas also with assessing genetics. So we can stratify cells by carriers and non-carriers of a particular variant, or use allele-specific expression. I come back to that later. Actually, we use ASE mostly for these types of analysis. Because you have an internal control for the same cell, you can use the whole transcript to understand where is that cell on this axis. And you can use the allele-specific information to understand, is there a genetic effect? Is there a difference as a function of genotype? I just want to jump right in and how these types of results look. So here you see a selection of a couple of hundred genes ordered in rows. So different transcripts are in rows. And on the left-hand side, you see the expression dynamics. So here are all the genes that switch off across differentiation. So they're highly expressed in iPS, lowly expressed in differentiated cells. There's a cluster of genes that are on intermediate stages, and there's a cluster of genes that are switching on across differentiation. Now, what you can anticipate if you look at the genetic effect estimates, you get a very different picture. So this is you looking at genetic effect size estimates using allele-specific expression of eQTLs for these corresponding genes. And for some, you see concordance that genetic effects are most active where the genes are highly expressed. But we see all sorts of other complementary explanations. For example, here are genes, and here's a whole cluster of those, which have decreasing expression, but the genetics matters most where the genes are least expressed, which I think quite counterintuitive and very interesting to see this emerging. And this suggests that there's substantial complexity where gene regulation actually occurs in the course of such a differentiation experiment. And here, for example, this example, we clearly see that expression level is not indicative of where genetic effects matter most. We also see intermediate cases. Here's a gene PIGP, which has strong effects in intermediate stages of differentiation. So if you look at the trajectory of expression changes, there are differences according to allelic state, but they only matter in intermediate stages. The endpoints and the beginning are shared, which I think is quite exciting. And again, this is just a differentiation time course of 3 days. So it's a very, very compact temporal window we're looking at. If you more formally cluster all these different genes by their dynamics, expression, and ASE dynamics, there's very little concordance. It's extremely interesting to see the complexity of different patterns that exist in these types of data, genes that increase expression and activate, but genetic effects manifest in the reverse order and all sorts of different patterns that came to light. I want to have one slide here about the method that we're actually using. So there's quite a bit of work in this area. I cited here David Knoll's paper, which I think was one of the first to do this systematically. So what actually we do for this analysis, we take eQTL variants that we know, that we identify in population, and then phase heterozygous variants of these single cell data, and then quantify in an allele haplotype specific way, essentially, the expression level of the haplotype that's associated with the T allele versus the expression level that's associated with the A allele. That's doable with phasing, and then we can use this ratio to test for changes in allele-specific expression as a function of differentiation time. We can actually just plot this quantity. That's what you saw in the previous plot as a function of differentiation. We can estimate that in every single cell. So this is actually whole transcript single cell data. So we have sufficient power to do that. But we can also you can also ask for more complex interactions. We now can assess, are there other factors that we can measure in cells that are linked to expression changes? For example, I mentioned at the very beginning of my talk that we can estimate, using factor models, cell cycle states. We can estimate respiration state in cells. And we can ask for each one of these factors, what are the contribution of these different cell state variables in modulating the effect of a genetic variant on gene expression? And the answer is you see everything you can imagine. You see respiration changes. So here, there's non-respiratory cells, these are respiratory cells, and you can see, again, genetic changes happening, or G1S transition. And in global, more than a quarter of all the eQTLs show interactions. And I think it's a quite an interesting result, and it really suggests that we have to pay a substantial amount of attention in asking texts do regular variants act, and segregating just by tissue is certainly not sufficient. There are very fine-grained regulatory differences that can emerge in such systems. I just want to show you one example here. You can even go to higher order interactions. So what I'm showing you here is essentially cells with high pseudotimes. So these are endodermal cells, low pseudotimes, IPS cells. And then, the allele-specific expression as a function of respiration. So you can see that there are differences, sort of even, combinatorial differences in the way. Or here you can plot respiration and pseudotime in this 2D plot, and you see that there's no effect in this light blue population, but there is an effect of respiration modulating allele-specific variation in the other. And what we're now actually I think is in quite exciting areas to think about how to do this more generically. So we use our methods that, you know, estimate similarity of cells based on the whole transcriptome. Yeah, so that's an approach we developed a few years ago, where we estimate how similar are cells post the whole transcriptome and use this similarity as an environment or cell state variable, and then ask generally for interactions. And what this allows us to do is to predict in every cell the genetic effect. So what you see here is not expression level of a gene. So these are our iPS cells, intermediate stage, end stages, but it's actually a predicted, interpolated single cell genetic effect for OCT4. And so here you see essentially really a continuous scale without making any assumptions of whether that's differentiation or pseudotime. It's essentially just taking the unbiased genome transcriptome-wide similarity of cells and then pooling and aggregating to be able to make predictions of where the genetic effects occur, and you know, the opposite effects and sign flips and a whole lot of complexity to sort of be discovered with these types of data. Yeah, with this I would like already to finish. I just want to say these ideas of pooling iPS cells and you know, scaling human biology essentially by taking lots and lots and lots of individuals and putting them into one well is something we're scaling right now. There's a project with Florian Merkel and Daniel Gaffney. It's also a bioarchive where we now look at much larger pools, 20, 30 lines in a pool, 250 iPS cell lines. And we look at also 52-day differentiation protocol towards neurons. So it's much more complicated than endoderm with 3 days. And we're also able here to recover the canonical cell types, neuronal and non-neuronal cell types. And we're able to also map eQTLs across the different cell stages. And for example, what we find is that across maturation, if you look at these more 52-day aged populations, they become much more similar to primary brain. This is sharing of eQTLs with GTEx. In other words, you know, in vitro aging cells over 52 days really produces neuronal populations that are from a perspective of gene regulation not too dissimilar to primary tissues as we see them in GTEx primary brain tissues. And I think it really just shows that we can really use these types of technologies to scale up human genetics and using interesting cellular systems. Okay, and I think I'll skip this in the interest of time, and I'm jumping right to acknowledgements. So this is, I really have to thank the lab. It's been we've now moved to Heidelberg. There's actually a picture from our first lab retreat. The work I presented has been by many individuals the spatial work by Valentin Svensson with Sarah Teichmann's lab, the multi-omics work really led by Rika Argela-Guerre together with John Marioni and Wolf Reich, and the factor model work as well, Danila Bredekin and Ricard, and the differentiation, the single cell work by Anna Klumo, Mark-Jan Bonder, and Daniel Seaton, who's now left the lab, and Helena Kilpin, so many alumni who've been involved in this. And yeah, with this, I want to thank you for your attention, and I take questions.
Well, thank you, Ollie, for this really nice talk. There are a number of questions in the Q&A panel that I'll read out some questions of them. Feel free to enter more and we will select them. So first question is from Shuba Varshini. Can MOFA2 be used for metagenome and metatranscriptome analysis, for example, from gut microbiome data?
Yeah, I'm jumping right back. I mean, we have used these models for, I mean, in theory, they're generic, you know, so you could use them for such analysis. We've worked quite hard on noise models, primarily for binary and binomial observation and single cell data. There may be, and I think primarily for microbiome, that's an important component because of the specific dropout characteristics. There will be a need to perhaps tweak the observation noise model. So I'm cautious. In principle, yes, but I think there might be I think a need to think about better noise models for metabolomics and microbiome as well, because of this extreme zero inflation that you observe in these types of data. Yeah, I think it's an area of future work. Yeah.
So, okay, so while Ollie is figuring out how to stop the phone from ringing, I'll read out the second question. If the voxel size is large enough, can you couple the composition methods with spatial gene analyses to get a sense of both cell type density variation and spatial gene expression patterns. So basically combine.
That's an excellent question. That's very doable, and actually you can in these spatial transcriptomics assays, there's sort of two ways to look at this. The one is technology will fix it, and we'll soon have just really true single cell variation in general. The other view is that there is a mileage in deconvolving these voxels. I've shown you these sort of voxels here. They're extremely large, and there's some ongoing work which will come out soon with Omar's lab at the Sanger, where we work on deconvolution methods. The question is whether you want to deconvolve that relative to reference. Yeah, so whether you want to use conventional single cell RNA-seq to define cell types and cell states, and then align those back. And I think this is a fruitful exercise, and there's been also already quite a number of computational methods for this task emerging that I would encourage you to take a look at. And I agree that's useful. But you have still this question in the end. You could then actually operate spatial variation. You could test spatial variability on the level of cell type densities rather than expression level of individual genes. So I think in a way, these questions remain relevant also in this context.
So another question on MOFA, on the distributional assumptions that are made. So the question is it based on CCA or group factor analysis? So what are the distributional assumptions?
Yes, I don't have a nice slide here. Apologies, that's the result of copying. Maybe I do. Let me just give it one quick shot. Maybe I'm lucky. Doesn't look like it. The key assumption that we're making is to constrain. I mean, there are two assumptions that I think relevant. One of them, the distribution assumption of the observed data. So there we at the moment at present support Poisson distributed observations. So this is Poisson given a latent model that is actually normal Gauss. So it's like a log normal essentially model for the read counts, which is for many single cell RNA-seq data appropriate. Bernoulli and binomial, all supported for these epigenome data sets which have a binary nature or a ratio phenotype. But I think the more interesting question is what are the distribution assumptions to some extent on these different weight matrices W and Z. And here we put a hierarchical prior on the relevance of features for individual factors, which allows you to sort of come to these variance decomposition plots, which also actually encourages zeros in those, so that specific factors are not relevant for some views. And in the MOFA 2 version, what we've done is actually just generalize this in two ways, basically having an ARD prior on factors, but also for different subgroups of latent factors. Basically, there's an ARD prior in the factor space and in the loading space that tries to estimate what's the explained variance and the relevance of a specific factor in a subset of samples on a subset of loadings. And in addition to this, there's an element of sparsity in these specific weights. But we, for example, do not encourage non-negativity. So these values can be positive and negative. Yeah, I hope that answers it.
Also, feel free to raise your hand in the participants list. And Bill Noble has asked another question about whether it's possible in the pooling strategy to uniquely identify individual donors, or more why it is not possible, for example, using barcodes.
Yeah, it's a good question. I mean, the first answer is obviously identifying donors is doable. I showed you here because that was the method that we developed, was initially to link cells to subclonal structure. Yeah, but if you think about it, obviously the genetic diversity difference between two individuals is much bigger than such a subclonal structure. So deconvolving individuals is simple in comparison, and this has been demuxled and other methods that address the same tasks. And there's a whole zoo of methods that have become at a similar time. So what you can actually do is you can even cluster cells unbiasedly by their genetic makeup and deconvolve them without a reference. So you don't even need to know what's the genetic state of the individuals, although that's of limited use because then you have to still figure out which cluster is which person to be able to drive your analysis. So clearly you can deconvolve people, and that is useful. And you're completely right, one could use barcodes. It turns out that this natural genetic barcode is just extremely convenient because you don't have to take any further action. You know, it's a barcode with 100% efficiency. It's cheap, it's simple. And if you're interested in studying intra-individual variation in the first place, then obviously it makes sense to use this barcode. If you were pooling replicates pre-post treatment, this barcode would not be applicable, right? Because there's no difference in the genetic makeup between the same cell from the same person pre and post treatment. So it depends on the application use case. But for scaling and enabling these human genetic studies, I think it's an appealing approach. And there's a final, yeah, no, I think I'll leave it there. Thank you.
So there's one more question. Could it be possible that the importance of the genetic variation is associated with a specific repression of the gene expression? Example given from binding sites of the repressor TFs.
Sorry, can you just repeat this question? I didn't pay attention sufficiently.
So it's could it be possible that the importance of the genetic variation is associated with a specific repression of the gene expression?
I see. Yes, I mean, I think this is relating to this dynamic expression analysis. I mean, what we have found in many cases that in some cases, roughly a third of those associations, we could identify changes in enhancer makeup for distal regulatory variants at least, that could explain changes in the importance of these regulatory variants. So, I mean, it's clearly the case that all the canonical explanations do apply. Changes in enhancer marks and other elements. What we have had, to be totally honest, had limited resolution for is really pinpointing the specific causal variant of these eQTLs. And the reason is 125 people. So linkage blocks remain still pretty big, and and really being able to link these eQTL variants to specific enhancers that change across differentiation is somewhat challenging. Hence, I'm a bit careful. I mean, we have some mechanisms for some examples. I think we have them also in the supplement. But in several cases, we just don't know what is really happening, what drives these changes. But I should maybe also mention something that is maybe independent, but that's something also we assessed, which is a quite other exciting dimension, is whether cells differentiate at different speeds. And for example, we just have global effects that some carriers of particular variants, such as poorer differentiation factors and others. Oh my God.